Okay, good morning. So today I'm going to present some of the research from my PhD thesis, introduce you to some of the localities that I worked on and some of the fieldwork that I did, and a little overview on what I actually did for my thesis. So the overview of my talk, I'm going to go through a little bit about the Cambrian explosion, just reintroduce it to people if you've not heard about it before, and talk a little bit about the importance of the Burgess Shale, go into a little bit of the fieldwork that we did in 2010, talk about a new taxon, a new species that I described in 2012, go through the geology and preservation of the material at my locality, go into a little bit of the diversity and ecology at this site, which was the main aim of my thesis, and then to just briefly talk about some other new localities that have been found. So looking at the history of life, so most people here are very familiar with the Mesozoic and the dinosaurs and the Jurassic and Cretaceous, but I'm going to bring you right back to the bottom. Okay, so the Precambrian, before we actually had any major signs of life. The very first thing we find in terms of large fossils are the Ediacaran, and as you can see from the image here, these fossils do not look like anything that you would find on Earth today. So it's really not until we break through into the Cambrian, so about 542 million years ago, that we start to find animals that we can recognize in lineages today. So this has been earmarked evolution's Big Bang, or the Cambrian explosion, the Cambrian radiation, and that is because at this point in time we suddenly have this huge diversification of most of the lineages that we see today. So why did this happen? So there are some reasons for this relatively quick burst in life, and that potentially could be environmental changes. So for example, ocean chemistry might have been changing at this time that was conducive to life growing very fast. There might have been evolution of developmental genes. So for example, perhaps with the development of sight or some kind of arms race, it could have spurred animals to evolve faster. Also, ecological innovations could have played a role. So for example, we know that substrate was changing around that time, so perhaps it made it easier for animals to actually live in these environments. So how do we know about this huge radiation in life, considering that the majority of animals that we're getting preserved are actually completely soft-bodied animals? So there's very few vertebrates in this material. And that is because we have exceptional preservation from Burgess Shale type deposits that are now found globally. But the one in particular that I'm interested in is the Burgess Shale in Yoho National Park. And the reason that the Burgess Shale and other localities like that are so fantastic is because without exceptional preservation, really all we would have in the fossil record at this time is material like this. So for example, trilobites and brachiopods are invertebrates that actually have some kind of hard shell or exoskeleton, so they're more readily preserved. But the majority of invertebrates would not get preserved. So if we were to look at a reconstruction of a Cambrian community without the soft-bodied animals, you would have something like this. But with the inclusion of all these exceptionally preserved fossils, we can have a reconstruction that's a lot more clearer about the full picture of what was actually living in these environments at the time. So the Burgess Shale as a locality itself, it's in terms of fossils, it's incredibly famous. It was found over 100 years ago, and a lot of work has been done in that time describing material from the Burgess Shale. And why is it so important? It is important because there are over 200,000 specimens collected from this locality. So it is incredibly diverse with over 200 species described. And as I said previous, it has the first appearance of many modern phyla. And since the original Walcott quarry was discovered in 1909, there have been more than a dozen localities found around the area in the national parks. So this makes it very useful for looking at changes over larger geographic scales, and also temporally, because these localities are not all deposited at the same time. So this gives us an opportunity to actually look at the community assemblages in each of these localities, compare them with each other, and see if things are changing throughout the Burgess Shale and how this can compare to other localities globally. So how was the Burgess Shale preserved? So here is just a reconstruction of a carbonate platform, and below this carbonate platform in this mudflow area, what was happening was these communities were living and basically getting buried repeatedly just below this escarpment. So the cathedral escarpment is thought to play a role in the quality of preservation of these animals, or at least was traditionally thought so. So where is the original locality? This is Fossil Ridge in Yoho National Park. This is the Walcott Quarry, and just to show you where the escarpment is, so you can see the proximity of the quarry to the escarpment. And this is the quarry itself. If you can see all these striations, these are just individual layers, event beds. There's some background sedimentation here. The beds vary from a couple of millimeters thick to tens of centimeters thick. So there's a little bit of variation, but for the most part, there are communities just getting repeatedly buried in situ at this locality. And you can see all of this shaly material that's fallen off. Many of those slabs will actually contain fossils. So when we think of some of the more famous Burgess Shale taxa, often what happens is because the Walcott Quarry has been the most comprehensively studied of these localities, people think the Walcott Quarry is a typical Burgess Shale community. But so some of these more enigmatic forms, what I'd like to highlight here is that while Anomalocaris is incredibly abundant as a taxon throughout the Burgess Shale localities, there's actually only one individual claw found at the Walcott Quarry. So this was not a major component of this community. And also here, Santacaris is not found at the Walcott Quarry at all. So that was part of what I was trying to do in my PhD, was to look at, can we actually describe what is a typical Burgess Shale community? So here's a geological section of some of the main localities. Here, each of the stars represents a locality, and in blue here is the cathedral rim, or the escarpment. So you can see that we have time-successive assemblages. And this is nice because some of these assemblages are actually quite close together in time, so we can actually see these communities evolving through time. So in terms of paleocommunity analysis, what has previously been done, there have been a couple of studies that have been done on the Walcott Quarry and the Raymond Quarry, which is just above the Walcott Quarry, so close to the submarine cliff. But the only real comprehensive study has been Karen and Jackson in 2008. And the only locality that has been away from the cathedral escarpment, so actually above the escarpment, is the Stanley Glacier site that has a preliminary analysis published by Caron et al. In 2010. So for my PhD research, I was looking at another locality, the Tulip Beds locality. And this is a locality on Mount Stephen, so close to the Walcott Quarry. It was discovered by the ROM in 1983, and it has about 10,000 specimens. So these were already in the ROM's collections, so we knew that it was numerically important, but wanted to actually identify the community assemblage, and with the aim of answering these questions. So is the depositional environment different from the Walcott Quarry? So some people had suggested, because of the kind of material that we were seeing in the Tulip Beds, that it was different from the Walcott Quarry, that the community composition was different and the environmental conditions were different. So what I aimed to do was actually compare this with the Walcott Quarry, see if there are any major differences and what's potentially driving these differences. So here's just a close-up map. So for those of you familiar with this area, the town of Field is just on the bottom left of this map. The Walcott Quarry here is the green star, and the Tulip Beds locality is the yellow star. So geographically, at the moment, they're about 5 kilometers from each other as the crow flies. But stratigraphically, They're a little bit more distant in that the Walcott Quarry is part of the Walcott Quarry member of the Burgess Shale Formation, and the Tulip Beds locality is part of the basal part of the Campsite Cliff Shale member. So stratigraphically, so time-wise, they're a little bit further apart from each other, and in terms of what has been suggested before, Fletcher and Collins suggested in 1998 that the Tulip Beds was more distal from the escarpment, so that it was further away from the escarpment, and some people had suggested the preservation might not be as good because it was further away from the escarpment. So here is the Walcott Quarry and the Tulip Beds, and then to actually look at the locality on Mount Stephen, so this is the north side of Mount Stephen. The Tulip Beds is up here, you can see about halfway up the mountain, and just a close-up of the locality. It's not the most hospitable of places. It's pretty difficult to get to, and if I zoom in just a little bit further, you can see here is the only place that you can actually put a campsite at this locality, and the only way you can get to it is by helicopter. So in 2010, we went there to do some geological work, fill in some of the gaps, and as you can see, basically our supplies were just dumped on this tiny little limestone crag. And so the same place that the helicopter lands is also where we put our camp. So it's a very small enclosed space. It's difficult to tell from the photograph here, but either side of the helicopter there is just basically sheer drop. So you have to be really careful when you're walking around this campsite, especially when the wind is blowing. So here is the camp all set up. This picture is taken from the quarry site, so one of the sub-localities here. And you can see the shaling material that we have to work with. And again, if you turn around just at this locality and if you walk the wrong direction, you have a very steep drop. So you need to be really careful. And the other sub-locality is a couple of 100 meters away across this scree slope, which again is incredibly dangerous. So in 2010, we actually had a professional climber with us who roped off this section, so we were able to tie in as we were crossing. And it has amazing views because of where we are on the mountain. It's stunning. So you're looking right down the town of Field here. But the scree slope itself is actually quite dangerous. And on this sub-locality, so on the talus slope, it's really difficult to get to the rocks that are actually producing all of this scree. So essentially all of the collections from this side of the locality are just from talus material, loose material. So we didn't actually know where the material was coming from. But at the other site, the quarry site, we were able to do some logged sections for more detailed work and also try and look for in situ fossils in 2010. So this is one of our sections before it was cut out. So you can see we just marked them up, and basically this was the first attempt at actually looking at any of the small details of this locality. And of course, like all paleontology work these days, you have to be very safety conscious. So whenever we were using tools, we had to have eye protection, we had to have face masks if we're using the rock saw, and ear protection. Of course, this pretty much results in you not being able to see well, hear well, or breathe well, but you're pretty well protected. But for the most part, the tools that we're using constantly at this locality are a chisel, a hammer, and a deck scrub. So pretty much for most of the time, we're just going through loose material, just basically cracking it open with a chisel, hoping that there's something fantastic inside. More often than not, there isn't. But sometimes you get something spectacular. When you rub it off with your deck scrub, you can see some stunning material. So this is Martin and Mishi, two of the guys that were working with us in 2010. And here is the end of the fieldwork, so as we're putting the camp away. And you can see here in the foreground, all of these white burlap bags are filled with the fossils that we have collected. So it's a little bit different from dinosaur research in that we can probably fit 20 or 30 fossils in a tiny bag and then just pack the bags into a chest, and then the helicopter brings them down. So it's much more simple to collect lots of material. But I just want to show you just a couple of pictures of if you've ever been to the mountains, you know how changeable the weather is, and especially on this locality, it wasn't pretty when the wind starts howling at night. But you can see our tents were held down by rocks because there's really nothing to tie into this locality. And the only real bad weather we had at this locality was some rather large hailstones. But I'm not going to go into the details of the rest of the fieldwork we did in 2010, but at one of the other localities, we did come back one evening to find our kitchen tent completely flattened. So it took a lot of Gorilla Tape to put that tent back together. And then here you can see Martin having to retrieve his tent, which had blown across to the other side of the cirque. It was in multiple pieces. And if it wasn't for the fact his clothes were in the bottom of it, it probably would have flown away entirely. And you can also see here at the front that it busted through the bare fence. So we had to have bare fence at this locality, and our kitchen tent was separate. But at this stage, there was no fence left. Also, again, it can be sunny in the morning. It can snow by lunchtime. And we did have some snow, which didn't impede us too much. It melted fairly quickly. But it was a little bit difficult when the helicopter came to take us out during a snowstorm. But it could have been worse. So this is an image from the Walcott Quarry in 1999. In this particular field season, they spent about half the field season actually digging out the snow first before they could actually get near the rocks. So in terms of weather, we had it pretty easy in 2010. So now I'm going to move on to actually talking about what I did with the material that was collected. So the field collections range from 1983, when the locality was first found, to 2010. It contains 2017 slabs, of which 9,218 specimens were identified. We did some geological logs and sedimentological analysis, and this is work that was done with a sedimentologist from California. But the first thing I did was actually to describe this animal from this locality nicknamed the Tulip Animal. So here's a reconstruction that we did of the animal. Hopefully the name makes sense. But the reason that it was important to do this study first and foremost was that this taxon is so abundant at this locality. It's incredibly large by Burgess Shale standards. It was cryptic in that it wasn't readily identifiable as anything or assignable to a particular group. So for example, if it's a new species of arthropod, we at least know that it's an arthropod. But in this case, we weren't sure what it was. It is unique to this locality. So again, it had some important implications for the ecology and community assemblage here. So I call this Cyphus actum gregarum, and it was published in 2012. And essentially, the publication was based on 1,525 specimens. So for any of you who work on dinosaurs, you probably know this is like a pretty large sample size. It ranges in size from about 2 to 25 centimeters. It is completely soft-bodied. It has a bulbous calyx, this long stem, and a small holdfast, and is interpreted as a stalked filter feeder. So morphology-wise, so here is a longitudinal cross-section and then just a cross-section through the calyx. And if you can see here, essentially it has this large bulbous calyx at the top. In the center is the stomach, which is completely surrounded by these lateral elements, which are interpreted as filter-feeding elements. And the stomach actually grays upwards, so excretion would have been at the top of this animal, which is quite common with filter-feeding animals. So just to show you some pretty pictures of the fossils, here's just some of the variation that we see in the holdfast. So it's pretty small, so this would have been the anchor point for the animal. As you move upwards, it has this long stem that has two parts, so the internal part was more rigid and less prone to decay. So even in the worst preserved specimens, the inner part of the stem was always preserved. And then into the calyx, you can see here there's some variation in the shape of the calyx at the top. Okay, so this crown on this animal could be quite narrow at the top, or it could widen out. And there are a couple of reasons for this. One could be taphonomy, so the way in which it's preserved or the angle at which it's found can change the shape at the top. But I also consider this to be somewhat biological. So in the ones that are preserved completely flat, they do show this variation. And this is what I think relates to its filter feeding ability, and it's all enclosed within a sheath. So I think this is an active filter feeder actively pumping water through the calyx. So in terms of size, so there's two here, but the little one is about 2 centimeters. And they basically have the same shape and general features right the way up to the largest one. So this one here is about 25 centimeters. And for those of you not familiar with the Burgess Shale, 25 centimeters is pretty big. And we know that this isn't complete because the slab is broken off at the end. So just to compare this, the largest complete Anomalocaris specimen that's been found is about 20 centimeters. Okay, so this is pretty big. In terms of biological affinity, unfortunately, this basically drew a blank. So this went into the Problematica group. And that is because stalked filter feeders generally have very similar features because of their lifestyle. But yet, because of the way they live, it's very difficult to actually assign them to a particular phylum. So here are two other stalked filter feeders from the Burgess Shale for comparison. But it is unlikely that Cyphus actum is closely related to any of these. But in terms of ecology, we were able to establish a little bit more. So here is a large slab, and it shows the gregarious nature of this animal. So just to show you an interpretive drawing of this slab, you can see here all of these specimens just basically overlying each other, and there's little sediment between these, so they're pretty much in the same position as they would be in life, except upright. So they were living in large groups together, but they weren't connected to each other. And while this animal was probably sessile for most of its life, there is the suggestion that it might not have been sessile all the time. So here is showing a close-up of two of the holdfasts, and you can see one is quite bulbous, whereas the other is flatter and has a grainy texture. And this might be indicating that these animals actually had the ability to move themselves around in the sediment. And in terms of how it filter fed, this is the way I have interpreted. So there are small holes at the base of the calyx, and it's likely that water was being pulled upwards through the pumping action of the calyx, with food being pulled out on these little striations at the side, and then down to the stomach, and then up the top. So in terms of what we know about this animal, it's incredibly abundant at this locality. It has an unusual ecological mode. There are very few active filter feeders found in the Cambrian, and it is of unknown affinities. But the major question that I had at the end of this study, and this was before I had done significant field work, was about its stratigraphic reoccurrence. So essentially I wanted to know, were all of these tulip animals coming from the same layer at the quarry, or were they coming from multiple layers at the quarry? Especially because this animal is not found at any other Burgess Shale locality. So looking at the geology of this locality, some of the questions that I had was, is the depositional environment different from what we know at other Burgess Shale localities? And where does this locality lie relative to the escarpment? So was it as far as Fletcher and Collins had suggested in 1998? And where do the fossils originate? So are they found in multiple layers, or are they all from one layer? So to go back to an image of the locality, just to draw up where the major units are here. So as the material where the talus and the scree is coming from is the quarry site and the talus site. This is just at the base of the campsite Tulip Shale member. And the quarry site, we were actually able to do some sections. So as you can see, it's not the easiest place to work in. So Jean-Bernard, my supervisor here, is actually tied into the section while using the rock saw. But we tried to take 3 sections. Section 2 and Section 3 were partially overlapping. So this was to look at where the material was coming from and what kind of geological features we could see within it. And this is a stratigraphic section that was done with Bob Gaines, the sedimentologist that we work with. And as you can see right up here at the top, the quarry site is sampling about 3 meters of this claystone. The talus site is sampling the same 3 meters, the lateral equivalent of this, plus about 3 meters more. And in terms of what is this rock type, it's thinly bedded, event-driven calcareous claystones throughout. So here's just an SEM showing the random orientation of the clays. And here's just a polished section of one of the slabs. And up here at the top, hopefully you can see these just really thin millimeter-sized beds. And there is some evidence of slumping or deformation before the rocks were completely solid. So here there's some slumping, and then if you look at the 2-meter section, which we actually logged in fine detail, you can see that this occurs a couple of times throughout the section. So what we did with the material that was not used for the geological sections, so with that it was cut, polished, x-rayed. The offcuts were split, put into 5-centimeter bins essentially, and tried to identify was there any fossil material in it, and if so, what was this fossil material? So here I've just binned it into 5 centimeters. This is just showing the major taxonomic groupings. And as you can see, there is basically fossils in every layer that we sampled. So the layers here that don't have fossils beside them, it's not because they were necessarily barren, but it's more likely that we just didn't have enough to sample. So in some of the sections, we didn't have any offcuts. So as you can see, it's a relatively diverse assemblage, and it's recurrent throughout. And in terms of depositional setting, yes, it's most likely that these were there were mudflow events that were coming in, basically smothering the entire community where it sat. But again, further from the escarpment. So the escarpment is up to 1 kilometer away here. So because of faulting, it's difficult to say exactly where it is, but we know that it is much more distal from, for example, where the Walcott quarry was deposited. So just to summarize the main geological conclusions, we know that it was further from the escarpment. It is a more distal setting. The beds are much thinner. We don't see the tens of centimeters that you see at the Walcott quarry. We have no evidence of background sedimentation. So this is different from the Walcott quarry, where you do see some significant background sedimentation. There is Burgess Shale type preservation throughout the layers that were sampled. And we do find taxa in many successive layers, including Cypress actum. So we know that the tulip animal was recurrent throughout these layers. So in terms of taphonomy, so this is the way in which things get preserved. Some of the questions that I had was, how are the taxa actually being preserved? Are they articulated or disarticulated? Have they broken apart? Is the preservation poor or excellent as you find it at the Walcott quarry? Are all the layers preserved in the same way? Are there differences in the preservation? Are the fossils actually in situ? So are they being preserved in the community that they would have lived? And are there any potential biases? So I looked at size and biomineralization as a potential bias that could basically cloud my data set and implications for diversity. So in terms of preservation, it's quite typical Burgess Shale type preservation with 2D carbonaceous compressions. So you can see here this slab is beautiful. If I show you another one, here it basically gives you an idea of some of the range of fossils you can find in just one large slab. So for example, here there are multiple claws from the large predator Herdia. I also have some Anomalocaris claws, so another of the large dinocarids. There are various kinds of sponges on this slab, as well as algae and cyanobacteria. So this slab shows the range of animals that you can find on a slab, as well as the range of sizes. So everything from tiny little individuals to large individuals can be found on a single slab. But not every slab has that kind of diversity. So for example, here is a really big slab of nothing but Anomalocaris claws. So basically the appendages of this predator, which possibly indicates that these were sitting on the surface for a while before they were buried. And the claws are probably the most resistant part to decay of these animals. And as you can see, they're just in hundreds here. So in terms of disarticulation, how we find animals, so here is Wawaxia, a type of mollusk. On the left here is a complete specimen. So it is All of the spines, all of the sclerites are still on this animal. If we move across to the right, you can see there's just some spines and some sclerites left, but they're still associated with each other. And then on the far right, there's just one individual sclerite. So this I can identify as a Wiwaxia, but it is completely on its own. There's nothing remaining of the rest of the animal. So some of the other things that we see at this locality, so for example, this is a molt of a trilobite olenoides, but what's really nice about this specimen is that although it has disarticulated and start to break apart, you can actually see that the pygidium, the tail region, is still associated with the head region. So this suggests that they haven't been transported very far. So other things that we find quite common amongst Burgess Shale localities are that these very large animals, like Herdia or Anomalocaris, it's very rare to actually find a complete animal, but it is quite common to find their individual parts in the collections. So for example, here you could have large gills, there's different kinds of carapaces, there's different claws and mouth parts that you can find, and for the most part these are identifiable to an individual species. So while most preservation is 2D carbonaceous compressions, there is some pyritization. So for example, this beautiful sponge. Also 3D preservation. So here in this brachiopod and preapulid worm, you're seeing two different kinds of 3D preservation. So while we have pyrite at the top, at the bottom here, you see the gut tract of this worm is getting preserved, and this is reasonably common amongst the worms and the arthropods. So the question about whether these animals are actually in situ, living in their home environment, I would say yes, they are. They are, for the most part, benthic animals, so animals that would live close to the sediment. There's no particular orientation in the large slabs, so if animals have been transported long distances, you would expect to see some kind of lineation in their bodies. The animals tend to be mixed sizes on a slab. There are some very large animals that are in some very small beds, suggesting that sediment has just been dumped directly on top of them. And often, like in this example here, you get individuals overlapping each other with no sediment between them, suggesting that they weren't moved very far to have lots of mud come in around them. So one of the potential biases that could be in my data set is size. So for example, it might be possible that you have an overrepresentation of larger species just because they have larger elements that are more readily identifiable in the collections in comparison to some of the small ones which might be very difficult to find. So I did some analysis on this and was able to show that abundance was not significantly correlated with size. So therefore, the larger you are, you're not likely to have an overrepresentation in the data set. And the size distribution is normal, so it's a size distribution that we would expect in a biological data set. And it is similar to what we find at the Walcott Quarry, so opening this up for comparison. Another potential bias is hardness. So for example, we know that there are lots of soft-bodied animals here, but are you more likely to get preserved if you have some kind of biomineralization, so some kind of hard part? So here, 42% of the taxa have biomineralization, but of that, there is no correlation with abundance. So you're not more likely to be represented in the data set if you have some kind of hard part. So for the summary for the taphonomy, the preservation is very similar to the Walcott Quarry, predominantly 2D carbonaceous compressions, some 3D pyritization and phosphatization, but the fossils are preserved close to their living environment. They have a range of, so basically we're sampling a range of taxa that were living or decaying in this area prior to burial, and there are no clear taphonomic biases that would impede further comparison with the Walcott Quarry or other Burgess Shale localities. So in terms of the diversity and ecology, which was essentially the main aim of my thesis, the questions that I had here was, what is the diversity and abundance of the tulip beds? What is the ecological structure of this assemblage? And how does the diversity in ecology compare with the Walcott Quarry? And if there are differences between these localities, what is driving those differences? So in terms of my data collection, I made a very large relational database looking at identifying each of the individual slabs and then all of the components on those slabs. This contained 2,017 slabs and 9,218 specimens that I counted. The taxa were identified to generic level where possible. So this is to try and avoid oversplitting. And in my data set, I did not include isolated elements. But for individuals that are only ever found as isolated elements, I tried to do a minimum count for those individuals. So for example, I know that Anomalocaris claws, each Anomalocaris has 2 claws, so I divided the number by 2. Also, algae and cyanobacteria, they're incredibly difficult to actually identify as a single specimen, so they were just counted based on presence on a slab. And the Walcott Quarry data was modified from the data used in Carroll and Jackson 2008. So in terms of data sets, although I have a really big data set, you can see it's absolutely swamped by the Walcott Quarry just because there's so much material being collected from there. So this is in terms of overall abundance. But in terms of diversity, I think the Tulip Beds is really holding its own here. So diversity, this is looking at the number of individual taxa. So here at the genus level between these data sets. So here you can see that 68 taxa are found at both localities, with 42 only found at the Tulip Beds and 63 only found at the Walcott Quarry. So I'm first going to go through the relative diversity per phyla. So this is basically grouping all of those taxa together into their individual larger groupings. And this is just looking at the number of taxa. So here you can see for both the Tulip Beds, which are the bars without the lines around them, and the Walcott Quarry have lines around the bars. You can see by far the arthropods are the most diverse groups within these assemblages. So this is followed by the porifera. So you can see there's some beautiful, stunning sponges found at the Tulip Beds locality, which are followed by the algae and cyanobacteria. And what's interesting about this is that many of the algae and cyanobacteria are unique to this locality at the Burgess Shale, but are actually found at other localities worldwide. So, for example, this one on the left, this beautiful branching algae, is found at localities in Russia. And then following on from that, the unknowns, so the taxa of unknown affinity are all grouped together as one group. And you can see here that there's a relatively high diversity in both the Tulip Beds and the Walcott Quarry. And at the Tulip Beds, I have some very bizarre taxa of unknown affinity, including the Tulip Animal, but also I have a very large assortment of blob-shaped things. So spherical fossils that I have no idea what group to even try and put them into, so that was not part of my study. But in terms of comparing this with the abundance, so on the left here I was looking at the number of taxa per phyla. On the right here I'm looking at the total abundance, so how many specimens were in each of these groupings. And you can see that while the one on the left, they track each other pretty well, it's on the right they start to fall apart a little bit. So here you can see that for the unknowns, so that includes the spherical things and the Tulip Animal, they are incredibly abundant at the Tulip Beds, whereas the Walcott Quarry is absolutely dominated by the arthropods in terms of abundance. So here is just looking at the subsampling. So with data sets that are vastly different in terms of size, rarefaction is a way to visualize the data so that you can actually compare more easily between them. So here, looking at subsampling, I'm going to just zoom in on the earlier part of the graph. You can see that the Tulip Beds is actually tracking the Walcott Quarry pretty well in terms of richness, so diversity, the number of taxa, and it has not reached a plateau. So it would be expected that perhaps with more sampling you would find more species at the Tulip Beds, whereas the Walcott Quarry has been pretty well sampled. and breaking this down into larger groupings. As you can see, the arthropod and porifera, so the sponges, both at the tulip beds, have a much steeper trajectory. So essentially what's happening here is that we haven't found all the arthropods or all the sponges yet at this locality. But where there is a big difference is with the unknowns, where the tulip beds, there are actually a huge abundance of unknowns, but they're not very diverse. So it's just a couple of taxa that are really driving this, this pattern that we see here. So in terms of ecology, I wanted to identify basically the mode of life for each of these taxa. So looking at the tiering levels, the feeding types, and their mobility. I base this on published data where possible. So essentially what I mean when I say tiering is, so for example, with a modern community, you have animals that live free-floating in the water. So these are planktonic. You have the nectonic animals that are free-swimming, so for example, the fish. You have nectobenthic animals, which are swimming close to the sediment for most of their life. We have epibenthic taxa, so these are taxa that live on the sediment. Endobenthic taxa live within the sediment, and then I had a certain amount of unknown, so I just did not know what tiering level they were. So again, here, each of these graphs are comparing the tulip beds to the Walcott quarry, both in terms of diversity, so the number in each of these, the number of taxa in each of these tiering levels, and then also abundance, so the total number of specimens in each of these categories. So you can see for both, they are very diverse in terms of epibenthic fauna, and in terms of abundance, it starts to break down a little bit for the nectobenthic fauna at the Walcott quarry, with a lot more unknowns at the tulip beds. And in terms of feeding, so here at the bottom, it ranges from suspension feeders to carnivores, the deposit feeders, the ones eating detritus, the primary producers, and the herbivores. Again, you see at the top the diversity, they track each other quite nicely, both dominant and suspension feeders and carnivores. But at the bottom, again, the tulip beds has a lot more in terms of suspension feeders, and the Walcott quarry is absolutely dominant in terms of deposit feeders, which you see in very low abundance at the tulip beds. So here I'm looking at motility, so basically an animal's ability to move. So some taxa were completely mobile, others are attached to the sediment for most of their life. And here again you see the diversity, they track each other really nicely, but in terms of abundance they start to break down. Okay, so you can see the Walcott quarry has a lot more mobile taxa, and the tulip beds have a lot more sessile taxa. So I'm going to go back to this graphic that I showed towards the start of the diversity section. So looking at the shared taxa and the endemic taxa, so the taxa that are only found in an individual locality. And I'm basically going to break this down into all of these individual taxa. So I don't expect you to be able to read this. There are far too many lines and far too many colors. But there's a couple of key things I want you to get from this graphic. So essentially what we see here are the ones in the middle are the taxa that are shared between the localities, but looking at their relative abundances. So for example, here, while we have one individual specimen of Anomalocaris at the Walcott quarry, it is 1,000 times more likely that you will find Anomalocaris at the Tulip Beds locality than the Walcott. And likewise, if you go down the other end, Morella, this little arthropod, is found in thousands at the Walcott quarry, but it is pretty rare at the Tulip Beds locality. So this is just looking at how the taxa compare to each other in terms of abundance. And then again, just grouping them in their phyla. This is just a little simple graphic showing which phyla are more likely to be shared between the localities and which are more likely to only be found at one locality. So for example, the brachiopods, sponges, the mollusks, the cnidarians, and the arthropods are more likely to be found at both localities. So just to summarize what I actually found in terms of the assemblage at the tulip beds. So you can see here, this is just a little graphic just showing what I think the community would have looked like. It was a diverse, for the most part, well-sampled assemblage. it's dominated by the arthropods and the sponges. And ecologically, it's dominated by the epibenthic and suspension feeders. So these animals that are actually living on or very close to the sediment. In terms of abundance, so the number of specimens that we find, they're mostly taxa of unknown affinity, followed by the algae and the arthropods. And ecologically, again, we have a lot of epibenthic suspension feeders and very few deposit feeders. But if we compare this to the Walcott quarry, for the diversity, it's very similar. Okay, so it is well sampled, it's dominated by the arthropods and a lot of epibenthic fauna. But when you compare the abundances, here's where it starts to break down, where there are a lot more mobile taxa and deposit feeders in the Walcott quarry and a lot more arthropods than you see at the Tulip Beds. So in terms of the community comparisons, so the localities have very similar diversity patterns, but differ greatly in their abundance patterns. And this is followed also in the ecological modes. So what is actually causing these differences, the breakdown between diversity and abundance? So it is possible that some of this may be due to the influence of one or two very abundant taxa in each of these data sets. Okay, so they're just skewing the data in one direction or the other. But it is probably also showing some subtle differences between the environment. So we know that the Tulip Beds is a much lower energy environment. It's also possible that there were some oxygenation differences between the two, with the Walcott quarry being better oxygenated than the Tulip Beds locality, but it's difficult to tease these things apart. So in terms of wider comparisons, where do you go outside of the Burgess Shale? There are some quantitative studies that have been done on material from China, the Xinjiang deposits. I did do comparisons between these localities, but I'm not going to show the results for these, but I'm just going to talk about some of the conclusions. So the community structures and the ecospace usage does not change significantly when you look outside of the Burgess Shale. It doesn't seem to change throughout the Cambrian. But I also want to say that using any particular locality, so for example the Walcott quarry, as being representative of a Cambrian community is problematic, especially if you're using the abundance data, which is what we are aiming to do at the moment in terms of ecology. We're trying to get to that point of being able to compare abundances. And further quantitative studies of Burgess Shale type localities will help with our understanding of regional variations of these communities. So I'm just going to finish with some other new exciting things that might lead to some further work. So previous ecospace usage, so how animals are using their landscape, it has been limited to biomineralized taxa, and that's because it's so difficult to track these communities through the Cambrian and into the Ordovician. However, new discoveries of Burgess Shale type preservation, for example, here in the Ordovician of Morocco, will allow us to actually compare soft-bodied community with soft-bodied community. And for those of you that spotted this in the news this week, there was a new Burgess Shale locality just published on this week. This is Marble Canyons, so in Kootenay National Park. And this is being heralded as possibly the closest thing to the Walcott quarry in terms of exceptional preservation and number of species and number of new taxa. So after a very limited field program, they were able to show 50 new species, and 10 of these are new taxa. So this really has some great potential for further quantitative work in the future. So I want to thank you all for listening to me for so long, and I also want to direct you towards the Virtual Museum of Canada if you have any interest in finding out more about the Burgess Shale, any of the animals. This was a project that I was involved with while I was in Toronto. This is an excellent resource for learning anything about the history of the site or the description and reconstruction, and there are lots of videos of the reconstructed environment. And I have lots of people to thank for this talk as well. Thank you.